Hello students, here we are in Biology 1. This is Module 5B. I have the picture up right here of a experiment that's in your book. It's called Experiment 5.2. I believe that is accurate, still the name of it. And it's a nice one to set up. Okay, so if you have a couple of bottles, a couple of balloons, some yeast and some sugar, you can do this experiment easily at home, and it is a really good one. So by the time we get to the end of this presentation, I'll give you some more details on that. The reason I had it here at the beginning is because when I did this in class time, I usually started early because sometimes it takes a little bit of time to develop. Okay, so let's get into the meat of the rest of Module 5. Okay, so this is like I told you last time, Module 5 is, I believe, the most difficult module in the book, or one of the most. And so we'll be done with it after today. How about that, right? So Module 5A was quite long and quite involved on the whole photosynthesis situation. And today we're going to look at the other side of that equation. So we're going to bring this in here. So we understand all about photosynthesis now, right? That's happening in plants. And now we're going to see what cells that do not produce their own food, in other words, not autotrophs, heterotrophs. A heterotroph is a living creature that has to get its food. It doesn't make it. An autotroph makes its food. Mostly those are plants, but also some very interesting bacteria, namely blue-green algae. We talked about that last time a little bit, how they're providing the majority of oxygen. So last lecture was pretty long. I don't think this one's quite as long, but it is just as complex because it's going to go through those processes just like we did with photosynthesis. All right, so let's see what we're talking about. All right. There we go. Let me go back one. Here we go. All right, so photosynthesis is a chemical reaction, right? We're going to take carbon dioxide. We're going to take 6 of those. 6 CO2 plus 6 waters, 6 H2Os. We're going to use energy from the light of the sun because everything to do anything needs energy. All life needs energy. All living on the planet needs energy, right? And plants are going to make this beautiful thing. They're going to make C6H12O6. That is, it says sugar here, but it's technically glucose, the type of sugar, and 6 oxygens, 6 O2s. When you get to chemistry, you'll understand that oxygen, when it's by itself, like it is here, it travels around as a group of 2, like they're twins, if you will. It's called a homonuclear diatomic. You might remember that from physical science. So we have 6 of those. If you were to count all the carbons on the left side and all the carbons on the right side, they would equal. They're easy to see, actually. We see we have 6 carbons here, we have 6 carbons here, right? Remember, the 6 here applies to the entirety. If the 6 is in front, it applies to everything. So there's 6 O2 here, 6 O2 here. We have 6 H2 here, that's 12. 12 here. We have 6 oxygens, we have 6 oxygens. So it balances out. So if we take this equation and we run it backwards, I'm going to put that next arrow in there. I'm not sure why my advancing isn't working. Let's see. Hold on. Let me use this. There it goes. Hopefully it'll stay up there. The arrow's going the other direction because this on this side over here is what's happening in you and me, okay? And other heterotrophs, most other heterotrophs. There are some that don't use oxygen, but let's talk about us for a minute. We eat food, including plants. That's what we'd find with the C6H12O6, and we're breathing. Right? So therefore, we're doing the opposite of photosynthesis, and that's what we're going to talk about today. All right, so you can see it's kind of two sides of a different coin, right? Two sides of the same coin. We have photosynthesis on one side that supports cell respiration, which supports photosynthesis, and back and forth, right? So the idea here is that I'm putting light energy, I'm putting energy into the system, and in my body, I'm getting energy out of the system. I can't get energy out if I didn't put energy in. It's just the laws of physics. Energy is neither created nor destroyed. It only changes form. So it's changing form from the sun energy to chemical energy. And the chemical energy is in the form of a chemical called ATP, which we mentioned last time, and we'll talk more about this time as well, standing for adenosine triphosphate. All right, so we call this cellular respiration, and you'll see the picture to the right here says aerobic respiration. Aerobic means that it requires and needs oxygen. Okay, there's different steps that take place, however, and some of the steps don't need oxygen. Okay, so we'll get more detail here in just a minute. Cellular respiration takes place in living cells. Okay, living cells, and that's almost all the living cells. There are a few weird things out there that do similar to photosynthesis. It's called chemosynthesis, but we're not going to worry about those guys right now. All right, in cellular respiration, glucose, which was made by plants, is broken down into energy, and the energy is called ATP. You can see right here, in each step along the way, we can see ATP was created in this step, and in this step. The vast majority of it's created in this last step, this beautiful, magical third step, called oxidative phosphorylation, or the ETC, the electron transport chain. So we have two types, like I said. We have aerobic, requiring oxygen, and anaerobic, that doesn't require oxygen. You've probably heard the word aerobic, maybe when it talks about exercise, right? Aerobic exercise, because guess what we're doing when we're breathing a lot, aren't we, right? We're getting our heart pumping and our lungs working, and so aerobic exercise means I'm going to increase my oxygen intake. So anaerobic means it's not. So now there are some bacteria out there that live in anaerobic environments. They are, and we'll talk about them later this year. They in fact, some of them are what we call obligate anaerobes, which means they die in the presence of oxygen. That's weird, right? They do, and so therefore they have to live in an environment that has no oxygen, which, as you can imagine, is a rare place to find, right? Way under the earth, in the caldera of a volcano, things like that. All right, so how do cells make ATP? That's the magic question, okay? This is the hard part right here. ATP molecule looks something like we're looking at over here. We're going to see these shapes over here. These are called aromatic rings. Every little spot right here has a carbon. Some of them are marked. There's a carbon with the oxygen sticking off, carbon, 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 carbon on each of these little areas with a nitrogen and hydrogen. So these are just showing how they're bonded together. The carbon's bonded to an NH2, carbon's bonded to a nitrogen. There's a double bond here, a single bond here, double bond, and then we can see how they're connected here, probably by a hydrogen bond right here, kind of magnetically, if you will. Here's our sugar, and then here's our magical group called the phosphate group. And this little, right here, this bond that's holding these two phosphates together right here has that energy. Like I explained last time, it's like a spring. If you pull the spring back, if you squeeze the spring and let go, it's gonna boing out on its own, right? It's going to use its energy that's been stored up when you squished it. You had to squish it, so therefore energy had to go in. That energy that went in to make this phosphate group, of course, would be sunlight. All right, here it is. Why is it so important? I'm simplifying the picture here by making my adenosine, all those rings right there, just calling it adenosine. And then here, I'm showing you that bond right there. It's called the high energy bond. When that bond is broken, lots of energy is released. Okay, it's like a little nuclear reaction going on there. All right, let's watch a video about it.
The structure and function of ATP. Adenosine triphosphate is an important molecule in the transfer of chemical energy within cells. It's often referred to as the energy currency of a cell, as it can pick up energy and transfer that energy to another chemical process. Cells produce ATP continuously, storing small amounts of it, but generally using it immediately. ATP is a nucleotide, as it contains a 5-carbon sugar, a nitrogenous base, and a string of 3 phosphate groups. Its nitrogenous base is adenine, which is also part of DNA and RNA molecules, and its 5-carbon sugar is ribose. Together, these are referred to as adenosine. 3 phosphates are attached to adenosine to form adenosine triphosphate, as you can see here. The squiggly line between phosphate group 2 and 3 is a high energy bond. When this bond is broken during chemical reactions, ATP becomes ADP, or adenosine diphosphate, and a phosphate is released. The energy released is used by the cell for cellular functions. The ADP and phosphate can, with the help of energy from food, be resynthesized into ATP, as you can see here in this illustration. So this process is similar to a rechargeable battery. You use the battery, or ATP, to provide energy for something. The battery drains down, the ATP breaks down, you recharge the battery, and you eat food to recharge ATP. And that be the big
All right, looks like that's the end of that video. Okay, so I don't know how you felt about that. I felt like it was fairly well explained right there. All right, so we see here we've got our glucose that we got from plants. We're breathing oxygen, and we're going to make some things, right? We're going to break it back down like in the photosynthesis reaction. We're going to break it down into carbon dioxide, into water, and into ATP. Okay, so we exhale carbon dioxide. We also exhale water. You know that when you blow on a window and you can write your name in there, right? Yeah, so that's water coming out. All right, we have 4 stages. Now, these are things you have to memorize, okay? And it's not hard unless you have a real difficult time memorizing. But there's just 4 things to remember, okay? All right, number 1, it's called glycolysis. Glycolysis. You might want to remember the word lysis means to break up. So what we're taking is we're taking something and we're breaking it up, okay? So we'll see those steps here in just a minute. Then we have part 2. Number 2 is called the formation of acetyl-CoA, also known as the oxidation of pyruvate. Some people don't include this as a separate step. In other words, some people say there's 3 stages. Our book has chosen to go with 4. I think that's probably the easier way to understand it and the better way to explain it. 3 is, you know, the scary thing, Krebs cycle, named after Dr. Krebs, of course. This is the one I mentioned last week a couple times, how it scares everybody. I had erroneously said it was part of that photosynthesis. It's the opposite of what's going on in photosynthesis. We call it the citric acid cycle here in the cellular respiration. And then the 4th, we have the magical electron transport system. Okay, that is where the majority of the ATP is made. All right, so we're going to look at each one of these steps and we're going to see where ATP comes from. Interestingly, to get it started, we have to put ATP in. All right, because, right, everything needs energy to go. All right, lovely. How about this right here? Yikes, right? This is a tough one. You can see right here, I want you to take a look. We're inside a cell. So this is the cytoplasm. This orange yellowish thing right here is the mitochondria, right? All right, so what's happening is something starts outside the cell. I have to put 2 ATP in. I'm sorry. Maybe that. I think 2 ATP do come out, but I had to put some in to get this started. All right, we're going to see this is happening right here. Then it's going to go into the mitochondria and go through the electron transport chain starting here. The citric acid cycle is going to break up the carbons. All right, so notice 2 ATP come out here, 2 ATP come out here. Then as they're flying through these little kind of tunnels for the electrons to go through this electron transport system, look how many ATPs come out. Wow, it depends on who you look at, but it's about 36 to 38. I think generally we say it's 36. All right, we're going to watch a video on this in just a minute. When I was in college, I had a microbiology course. It was near the end of my career in college there. And I was given a blank paper and had to write this all down. I had to write down where every NADH went in, where every NAD went in and NADH came out. I had to write all the names. Oh my goodness. It was a tough final exam for that class. All right, we're going to watch, this really boils it down very nicely with color coding and stuff like that. So take a look here.
Aerobic respiration starts with 1 glucose molecule and 6 oxygen molecules. They react in a complex 4-step process that I want to illustrate for you by means of this animation. The first step is called glycolysis, and the background of this animation will stay pink throughout that first step. In glycolysis, the glucose molecule is split in half, some of the atoms rearrange, and 4 hydrogen atoms are liberated from the molecule. In the end, this results in 2 new molecules called pyruvic acid molecules, 4 individual hydrogen atoms, and some energy. Notice that nothing happened to the oxygen molecules at the top. That's because this step is anaerobic, which means it doesn't need oxygen. Alright, let's move on to the next step of the process. The next step is called the formation of acetyl coenzyme A, and I'm going to color the background of the animation yellow during this step. Notice we still have 6 oxygen molecules up at the top. That's because they haven't been used yet. We have 4 hydrogen atoms from glycolysis, and we also have 2 new things, 2 coenzyme A's. Where did these guys come from? Well, they're not a part of what we consider the necessary components for aerobic respiration, because they're produced in one respiration and used in the next. As a result, the net effect is they're neither used up nor produced. They simply cycle through respirations. What do these guys do? Well, they go react with the pyruvic acid molecules. They cleave a hydrogen from the pyruvic acid molecules, liberating carbon dioxide, and they attach to what remains. The result is two new molecules called acetyl coenzyme A's, two carbon dioxide molecules, and two more hydrogen atoms. Although a lot of chemistry takes place in this step, no energy is produced. That's the end of the formation of acetyl coenzyme A. Now we're ready for the next step, the Krebs cycle. I'll keep the background of the animation green during the Krebs cycle. The Krebs cycle is the first step that uses oxygen. Three of the oxygen molecules that we need for aerobic respiration come down and interact with the acetyl coenzyme A molecules. This breaks the molecules apart, liberating the coenzyme A's, producing carbon dioxide and hydrogen. In the end, this produces 4 more carbon dioxide molecules, 6 more hydrogen atoms, 2 coenzyme A's, and some energy. Now remember, those coenzyme A's will be used in the next aerobic respiration, so we don't really consider them products. They're just going to get used up again. Alright then, we're almost done. We just have to get through the last step, which is called the electron transport system. During this step, I'll keep the animation's background blue. In the electron transport system, the 12 hydrogen atoms from the previous steps interact with the 3 remaining oxygen molecules. This results in 6 water molecules being formed. That's the end of aerobic respiration. So what did aerobic respiration make? Well, it made 6 carbon dioxide molecules, it made 6 water molecules, it made some coenzymes for the next respiration, and it made a lot of energy. In the end then, aerobic respiration simply takes a glucose molecule and 6 oxygen molecules and makes 6 water molecules, 6 carbon dioxide molecules, and a bunch of energy. Remember, even though they're listed here, we're not going to consider those coenzyme A's products because they're going to be used in the next respiration step.
All right, I like that one. That is the original writer of the Apologia books, and you might recognize his voice if you've been in Apologia. Okay, so here's the chart. So you may want to pause this video at this time and recreate this chart. I believe it's in your book. You can look at it in your book. So here's what's happening in stage 1, 2, 3, and 4. The most important thing, of course, would be where the ATP is coming from. Over here we have a 2 coming out, 2 coming out, 32. So 32, 33, 34, 35, 36. So we go with the 36 ATP. If you go through and look at some details, you might say, hey, there are 2 other ATPs. So some people include those in, some people consider those recycled, you know, that kind of thing. So for our book, we're going with the 36, so just stick with that one. Notice also we need to know where everything takes place, but that's not too hard because we have outside the mitochondria and the cell, and then all inside the mitochondria. Mitochondrion, of course, is just a singular form grammatically of the word mitochondria, which is the plural form. So that's all that means right there. Okay, so go ahead and pause this and maybe write these things down, and you can see what's been used, what's been made, and where the ATP comes out. Okay? I'm going to move on. All right, we have another fun video.
As this mountain biker heads up the trail, the breakfast he ate this morning is being burned to power his bike ride. His breathing rate increases as his leg muscles demand more oxygen to burn more fuel. Let's zoom down to where this fuel is burned, our cells. Here, the blood vessel on the left delivers fuel and oxygen to a single muscle cell. In cellular respiration, energy in fuel is converted to ATP, shown here as starbursts. Most ATP is made in the cell's mitochondria. ATP powers the work of the cell, such as contraction. Let's take a closer look at how ATP is produced from a molecule of glucose, our fuel. Only the carbon skeleton is shown to keep things simple. The first step is called glycolysis, and it takes place outside the mitochondria. To begin the process, some energy has to be invested. Next, the molecule is split in half. Now, the molecule NAD+, an electron carrier, picks up electrons and hydrogen atoms from the carbon molecule, becoming NADH. Keep track of the electron carriers. They play an important role by transporting electrons to reactions in the mitochondria. In the final steps of glycolysis, some ATP is produced, but not much. For every glucose molecule, only 2 net ATPs are produced outside the mitochondrion. However, glycolysis has produced pyruvic acid, which still has a lot of energy available. Let's follow this pyruvic acid molecule into a mitochondrion to see where most of the energy is extracted. As the molecule enters the mitochondrion, one carbon is removed, forming carbon dioxide as a byproduct. Electrons are stripped, forming NADH. Coenzyme A attaches to the 2-carbon fragment, forming acetyl-CoA. Coenzyme A is removed, and the remaining 2-carbon skeleton is attached to an existing 4-carbon molecule that serves as the starting point for the citric acid cycle. The new 6-carbon chain is partially broken down, releasing carbon dioxide. Several electrons are captured by electron carriers, and more carbon dioxide is released. The carbon dioxide that you exhale comes from the reactions of cellular respiration. 2 ATPs are produced by the citric acid cycle for each molecule of glucose. At this point, only a small number of ATPs have been produced. However, more energy is available in the electrons that are being transported by electron carriers. While the citric acid cycle starts another round, let's follow an electron carrier to the next step in the process. Electron carriers such as NADH deliver their electrons to an electron transport chain embedded in the inner membrane of the mitochondrion. The chain consists of a series of electron carriers, most of which are proteins that exist in large complexes. Electrons are transferred from one electron carrier to the next in the electron transport chain. Let's take a closer look at the path electrons take through the chain. As electrons move along each step of the chain, they give up a bit of energy. The oxygen you breathe pulls electrons from the transport chain, and water is formed as a byproduct. The energy released by electrons is used to pump hydrogen ions, the blue balls, across the inner membrane of the mitochondrion, creating an area of high hydrogen ion concentration. Hydrogen ions flow back across the membrane through a turbine. Much like water through a dam, the flow of hydrogen ions spins the turbine, which activates the production of ATP. These spinning turbines in your cells produce most of the ATP that is generated from the food you eat. The process you've just observed, cellular respiration, generates 10 million ATPs per second in just one cell. That ATP can power a biker up the trail, or it can power your brain cells as you learn challenging biology topics.
Yes, challenging biology topics, right? There we go. I like that one. I thought the animation was really well done. Okay, so let's see. Molecular modeling activity of cell respiration. I believe here there is an opportunity if you want to take some toothpicks and marshmallows or gumdrop candies and then take those apart and make them into the other things. I think it's in your book, so check that out and see if you see that. Okay, what is ATP and what is ADP? ATP is the energy package produced in cellular respiration and used by cells. Keep in mind that plants are doing this too, right? They're doing it at nighttime when there's no sunshine, so they're making ATP for themselves. Okay, ADP is when you take one of those phosphorus off of there, phosphates, I guess I should say, called adenosine diphosphate, general energy provider delivery service. I think I mentioned last time that a lot of times the ADP is just recycled back to ATP, but that second phosphate on there can be broken off with the high energy bond between there and the first one. So that can be done, so it can be a general energy provider. Okay, when ATP is transported to the cells, enzymes break the ATP down to ADP and a phosphate group. The energy is released in that high energy bond as it breaks, and it goes to the cell, and ADP and phosphate are then recycled. All right, so we also have a process called fermentation. So that is the production of ATP without oxygen, okay, an anaerobic process. You actually know about this even if you don't know that you know about this. Have you ever been on some sort of physical trek that you've been on, for instance, let's say you ran in a race. Okay, so you got yourself ready by practicing and getting your body ready to race and that kind of thing. And you go for, let's say, a 5-mile race and you're running for a distance. Well, the next day your muscles are so sore, right? Well, that's because you at one point had what we call an oxygen debt. You were not able to produce the amount of oxygen you needed to produce your ATP. So your body went into an anaerobic process of forming ATP. And the byproduct of that produces lactic acid. Lactic acid kind of collects up in your muscles and it hurts. So we know how that happens, right? So that's happening sometimes when we kind of overexert ourselves. I don't even really experienced runners, people who are in the Olympics, they'll have sore muscles too. There's just so much your body can do. But just make sure to keep breathing. The other process of fermentation, which you probably would have recognized from the word fermentation, is the formation of alcohol. Literally making alcohol, wine and beer and things like that can be made through a process of fermentation. All right, so here's what happens. In our glycolysis, we're going to see our glucose. We're starting over here with our glucose, right? We're going to go through this whole process of the NAD stealing the hydrogens and putting it on. Then we're going to take our ADP and form our ATPs converted into our 2-pyruvate. This is just one of those steps, right? Here we go. There's our NADH. Following the process here, we're talking about lactic acid fermentation right here. This didn't require any oxygen. No, there's no oxygen there, right? So I thought, okay, I guess that's not a video. We'll have a video in a minute. So lactic acid fermentation occurs in humans, all right? Our glucose, our ADP and our ATP right here. We're making our pyruvate, and we're going to go back when we don't have enough oxygen to proceed because we've been using it up. Then we're going to go back here and make this lactate, okay? No intermediate electrons accepted. All right, here's the video. This is going to be from that first picture we'll show you. Here we go.
Acid fermentation, as done by cells such as your muscle cells, for example. Well, your muscle cells can do aerobic cellular respiration. They can shift to lactic acid fermentation if they experience an oxygen debt. This could happen if you're working out very intensely, where your blood is unable to deliver a sufficient amount of oxygen to them for their demand. Just like alcoholic fermentation, we start with glycolysis. That yields the 2-net ATP, 2-pyruvate, and 2-NADH. But now we need the step to regenerate the NAD+, and this step is a bit different from alcoholic fermentation. The 2-pyruvate on the reactant side will ultimately yield 2-lactate. The pyruvate can act as an electron acceptor, allowing NADH to be oxidized to NAD+, so that glycolysis can start all over. By the way, this lactate product, or specifically its other form, lactic acid, has often been blamed for the muscle soreness that occurs after intense exercise. In many of my teaching years, this was often discussed, but there's some recent research that may contradict that. Science is so cool. Check out our further reading suggestions and our video details to learn more. Lactic acid fermentation is also done by bacteria that are involved in making yogurt, and lactic acid can contribute to its sour taste. So overall, fermentation is a pretty remarkable process. Although, it does make us appreciate oxygen because despite how absolutely awesome fermentation may be, it just can't make as much ATP as aerobic cellular respiration.
Okay, that's interesting about the acid not causing muscle soreness. I've of course had this video in here for a long time, and I've heard it before. I haven't read anything. I haven't seen anything about that. So I'd have to do a little more research to find out, what other process in there. We've been blaming lactic acid this whole time. Perhaps there's something new that's coming up, which is a beautiful way to remind you that science is never 100%, right? We think we know what we know, but who knows? There's something we may not know, and it may change everything. So always keep your mind open with science. And so someday, maybe if I'm interested enough, I'll go check out and see what the deal is with the lactic acid. Okay, I think that's the same one. Okay, sorry. All right, the next step, of course, the other type of anaerobic process is called fermentation. All right, the one that you might be a little more familiar with because you've probably heard of beer and wine. In fact, they're showing, a wine right here. Notice we have a yeast. I know, both of those, beer and wine, are made with yeast starters. All other alcohol, by the way, is stronger because it goes through a distillation process, a chemistry process. But it started over here with this stuff. It started with fermentation from a yeast, making glucose, and coming down and ending up with not going through the rest of the cycle over here, coming over here and going through making ethanol. Ethanol is just the name we use for alcohol. Ethanol is a type of alcohol, and it happens to be the type of alcohol that humans consume. Notice that we also have bread over here, right? That's going through a fermentation process too. It's just that the alcohol, what happens is during the process, CO2 is also produced, which makes bubbles, and the bubbles in the spongy bread make those holes in the bread. And when you cook the bread, it rises because of the bubbles, right? You've probably made bread before. That might be a fun thing to do for this module as well, make some bread. It rises up because it's making the bubbles, and when you cook it, when you bake it, all the alcohol goes away. In fact, the burning off of that alcohol is that delicious smell that you smell when bread is baking. All right, this is the video on this side.
Alcoholic fermentation is done by some types of yeast. So first glycolysis happens, which yields 2 net ATP, 2 pyruvate, and 2 NADH. Now we need this step to regenerate the NAD+, so we can keep doing glycolysis. The 2 pyruvate is used, which will ultimately produce carbon dioxide and 2-ethanol, alcohol. But the derivative of pyruvate shown here, acetaldehyde, can act as an electron acceptor in this process, so the 2-NADH can be oxidized to 2-NAD+, so that glycolysis can start all over. Since alcohol is a waste product in this process, this is why many yeast are used to produce alcoholic products. Yeast also can do alcoholic fermentation in making bread, and the carbon dioxide product we mentioned is involved with helping the bread rise. The tiny amount of alcohol produced in the short fermenting time of bread will evaporate in the baking process.
Okay, yeah, there you go. That's what I was telling you about the evaporation process. Okay, this is also a video. Remember, your module talks about this this lab. Let's see how it works out. You can see here the balloons are expanding.
Let's look at this experiment a little closer. Yeast is a fungus, and there are about 160 known species of yeast. Yeast is very small. 1 gram holds around 25 million cells and has been used for thousands of years to help bread rise. But how does a fungus help bread rise? It has to do with what yeast eats, how they metabolize that food, and what waste products are released. Yeast feeds on sugars and starches. When the yeast consumes the sugar in our bottle, it breaks it down to be used as energy. But like all living beings, waste products are produced when metabolizing food. When the yeast metabolizes the sugar, the waste produced is carbon dioxide gas and ethanol. The carbon dioxide gas floats out of the liquid into the bottle, producing bubbles, and eventually floating up into the balloon. As the yeast consumes more sugar and produces more carbon dioxide, the pressure inside the bottle and balloon increases. This pressure presses on the walls of the balloon, causing it to expand and grow. The equation for this reaction looks like this. The reactant sugar, sucrose, is on the left, and the products, ethanol and carbon dioxide, are on the right. The bottles with greater amounts of sugar have more food for the yeast to consume, which results in larger amounts of carbon dioxide produced and a larger balloon on the top of the bottle.
Okay, so notice he has the 4 bottles. You could just do the 2, one with sugar, one without sugar. Yeah, but it just shows you how that's going to expand that way. Because guess what's, he said it, the CO2 is being produced right there. Okay, so this right here for understanding what's happening during photosynthesis versus respiration and what is happening in both, all right? So you can see it's a little bit, I'm not crazy about this little chart right here because truthfully plants do respiration too. They do. So they kind of do everything. But animals do not. So animals do not do any of this stuff right over here. And we have some interesting like vocabulary words right here you can check into in your book right here. The numbers associated with these is because it came out of a lab book that had a special exercise that you were supposed to number them, but you don't need to worry about that. All right, here's another chart. I just wanted to throw this in here real quick. I hope it doesn't confuse you. Notice right here, we're going to end up with that 38. I just wanted to show you how sometimes people will count that second, those 2 other ATPs right in here and make it 34 instead of 32. So 34, 35, 36, 37, 38. Just know that sometimes there can be disagreement on that or just clarification in the sense that, where is that ATP going? Okay, what being recycled, that kind of thing. All right, so that is it, y'all. See, that was a pretty quick lecture. Wasn't as bad as last time. If you need some clarification in some of the things, go back and watch some of those videos. Maybe, listen and pause it and see if you understand what they said, back it up, that kind of thing. And try to get a hold of this. Just, bottom line is that you want to know that, what the 2 systems produce. So photosynthesis, of course, is using the energy from the sun to make glucose and oxygen, And cellular respiration is using glucose and oxygen to make ATP. That's the goal. There's the other stuff, of course. We're making water and CO2, which the plants can use, by the way. But the idea is to take those carbons, interestingly, right? Just take those, use the energy in those bonds, and rearrange them so we can have that phosphate energy, high energy bond, so that it will release energy. It's pretty amazing stuff. It is, once again, I would say, God showing off, right? The complexity of a simple chemical reaction. And as you know now, there's nothing really simple about it. Okay, so we've gotten through Module 5. Hooray! Next time we'll get together for Module 6, and we'll see what that is. And then as we move through the year, of course, we're going to get to the more interesting, the stuff you can see. We'll be looking at bugs and animals and, plants and things like that, microscope work and that kind of thing. Okay, students, have a great week, and I'll talk to you soon.